Hello, my name is Lauren Hill and I'm a biology major at the College of Worcester. Today I will be giving a quick overview of my senior thesis research surrounding the plant Echinacea angustifolia and its bee pollinators. To begin, I'll give some background on how plants reproduce. Plant reproduction can take many different forms, but the most common is sexual reproduction through pollination. Plants that reproduce through pollination have both male and female sex organs. When pollen from the male organs makes its way to the female organs, reproduction can occur. However, many of these plants are unable to self-pollinate despite having both sets of reproductive organs. This means cross-pollination is needed, in which the pollen of one plant is transferred to the female organs of another genetically different plant. In order to facilitate this pollen movement, most plants use pollinators, as shown in the diagram, the most common of which are bees. Though honeybees likely come to mind as a major pollinator, there are various bee species that are important to the propagation of plant populations. Understanding what pollinators are important for plant reproduction can help us understand how to better protect and conserve plant populations. In order to conduct my research, I used the study species Echinacea angustifolia. Echinacea has multiple species of bees that contribute to its pollination, but it is also limited in its reproduction due to the destruction of its habitat. In my study, I explore which group of Echinacea angustifolia's bee pollinators has the biggest impact on its reproduction by following the movement of pollen. In initial field studies, plants were designated to have their pollen collected by one of 6 pollinator groups. The offspring that resulted from this pollen collection and movement were then gathered for further analysis. By matching these resulting offspring to their parents, I was able to determine which pollinators are best for plant reproduction. In order to do this, I conducted multiple polymerase chain reactions, or PCRs, and microsatellite genotyping on the Echinacea offspring. Microsatellites are segments of DNA that are specific to individuals and can be passed down to their offspring. In this case, the microsatellites present in a plant are transferred through their pollen and found in their offspring. PCRs multiply these segments of DNA, allowing them to be analyzed and matched between parents and offspring. I ran a total of 37 polymerase chain reactions for 216 offspring and matched them with 225 potential parents. Some of my findings can be seen in the following figures. The top figure shows how many offspring were produced through pollination by each particular pollinator group. The bottom figure shows what percent of offspring were produced by each specific pollinator group for individual plants. My results showed that while some pollinator groups resulted in more offspring than others, specifically the Andrena pollinator category, the differences were not significant. This means no pollinator group was more beneficial than the others at increasing offspring production. My findings were interesting as previous studies found that when all bee groups worked together, which is demonstrated in the open pollinator category, there was a significant increase in the number of offspring produced. Despite these differing results, the final message is the same. Preserving pollinator bee species beyond the well-known honeybee is essential to protecting and conserving sexually reproducing plant populations.